Hello everyone, I'm back with another paper 52 video. Today we'll be solving Cambridge International AS and A-Level Biology Paper 5 Planning, Analysis and Evaluation. May June 2021 paper 52. In this paper we are going to discuss how to give a proper answer for this particular question paper and in general how to answer a paper 5 properly. And for those of you, a big shout out to you, those who are preparing for your exam. All right, and best of luck to your exam. All right, so let's continue with the video. Question number 1. The enzyme catalase catalyzes the reaction shown in figure 1.1. Hydrogen peroxide, broken down by catalase, produces water and oxygen. A student had read that copper 2 sulfate can act as an inhibitor, reducing the activity of catalase. So the copper 2 sulfate here, the student has learned that whether it can act as an inhibitor. Competitive, non-competitive, the question doesn't say that. The student wanted to investigate the effect of copper 2 sulfate as an inhibitor on the activity of catalase to test the hypothesis. So the student concluded the higher the concentration of inhibitor of copper 2 sulfate, the lower the volume of oxygen produced in the presence of catalase. State the independent variable and the dependent variable in this investigation. Since the student can change the concentration of copper 2 sulfate, so the independent variable will be the concentration of copper 2 sulfate. And the dependent variable will be the volume of oxygen produced because that is the result. Part B1. The student was provided with a 0.04 mole per dm cube solution of copper 2 sulfate. So this is the stock solution, we believe. All right. Describe how the student could use serial dilution to make a suitable range of concentrations to test their hypothesis. So the student, since the student has given, the student is given with 0.04 mole per dm cube copper 2 sulfate solution, the student can use serial dilution. The student wants to use serial dilution. All right, so the student can prepare a concentration. So the student, in order to since the student requires to make a serial dilution, in a serial dilution the student can prepare a concentration of 0.04 mole per dm cube, which is already given. So the student can then prepare 0.02 mole per dm cube, and then the student can prepare 0.01 mole per dm cube, 0.005 mole per dm cube, and 0.0025 mole per dm cube. Concentrations can be prepared. To prepare 0.02 mole per dm cube, the student can take 10 cm cube of 0.04 mole per dm cube copper 2 sulfate and 10 cm cube of distilled water. So after that, for preparing 0.01 mole per dm cube, the student can take 10 cm cube, 0.02 mole per dm cube copper 2 sulfate and add 10 cm³ of distilled water, and the student can repeat the process, so on and so forth, to make the other concentrations. Part 2. The student used the apparatus in figure 1.2 to collect the data needed to test the effect of different concentrations of copper 2 sulfate on the production of oxygen in the presence of catalase. 5 cm³ of 10% hydrogen peroxide we can see over here. Okay, we can see a mixture of catalase and a copper 2 sulfate, graduated tube to collect oxygen, and we can see water. All right. Describe a method the student could use to collect the data needed to test the effect of different concentrations of copper 2 sulfate on the production of oxygen in the presence of catalase. So we are asked to describe a method that the student can use to collect the data that is needed to test the effect of different concentrations of copper 2 sulfate on the production of oxygen in the presence of catalase. Now, our description of your method should be set out in a logical way and detailed enough for another person to follow. You should not repeat the details from B1 describing how to dilute 0.04 mole per dm³ copper 2 sulfate solution, or how to set up the apparatus shown in figure 1.2. Okay, so guys, I think this particular approach can be very good. You can follow, and I'll explain to you once I'm done with it. Okay guys, so describe a method that the student could use to collect the data needed to test the effect of different concentrations of copper 2 sulfate on the production of oxygen in the presence of catalase. Now the question says that our description should be in a logical way so that another person can follow. Now when writing an experiment like this, we have to understand that examiner necessarily doesn't want that we mention every nook and crannies of how to do a particular experiment. The examiner wants to know that what particular aspects of that particular experiment we're going to keep constant, what particular aspects of that particular experiment that we're going to change, how we are going to change it, how we are going to obtain the result, and the ones that we are going to keep constant, how we are going to keep it constant. And along with that, of course, we must mention about repeats, all right, the importance of repeats, and we must also mention whether there is any hazard or risk related to the particular experiment. So all experiments should be carried out at the same temperature using water bath. We know that temperature affects the rate of enzyme controlled reaction. So if there is a change in temperature, we won't understand the true effect of the copper 2 sulfate as an inhibitor. To keep it constant, all experiments must use the same volume of, to keep the pH constant, we must have the same volume of buffer solution in all of them. Because any enzyme controlled reaction, when the reaction carries on, it can change the pH of the particular reaction. So of course, using a buffer solution will be very will be very good, because it will not allow the buffer to have any effect on the rate of reaction. Same volume of catalase and same concentration of catalase should be used. All right, these are controlled variables. So same volume of copper 2 sulfate should be used. However, it's not same concentration, it's the same volume of copper 2 sulfate for each concentration that we are going to be using. The volume of oxygen produced in 5 minutes is recorded. So we have the time as a fixed, and then we only want to know how much oxygen is produced within that particular time. So if copper 2 sulfate is acting as an inhibitor, it is going to produce less amount of oxygen within that particular time. A control experiment with no copper 2 sulfate can be done for comparison. So to show that whether the presence of copper 2 sulfate is affecting the rate of reaction or not. Fresh solution of all reagent should be used for all experiment. All right, for each concentration of copper 2 sulfate, at least 3 replicates should be done. And as a precaution, catalyst, enzyme, hydrogen peroxide, or copper 2 sulfate are irritant, so gloves, mask should be worn for protection. Now, guys, one thing I believe is gonna help us if we include it is that about the control experiment, we said with no copper sulfate can be done for comparison. Yes, we can replace the copper sulfate with water. Part 3. The hypothesis the student tested was the higher the concentration of the inhibitor copper 2 sulfate, the lower the volume of oxygen produced in the presence of catalase. Complete figure 1.3 by adding excess labels and units, sketching a graph of the result you would expect if the hypothesis is correct. Well, if the hypothesis is correct, then we would get lower and lower volume of oxygen produced as the concentration of copper 2 sulfate is going to increase. So on the x-axis we are going to mention concentration of copper 2 sulfate / mole per dm cube. And on the y-axis we are going to have volume of oxygen. And since with increasing concentration the volume of oxygen is decreasing, so we're going to have something like this. Part C. The student carried out a further investigation to find out if copper 2 sulfate was a competitive or non-competitive inhibitor. The student measured the oxygen produced in the presence and absence of copper 2 sulfate at different concentrations of hydrogen peroxide. The student processed the data to find the rate of oxygen production. The student then plotted these rates on the graph shown in figure 1.1. Percentage concentration of hydrogen peroxide increases on x-axis. Rate of oxygen production is on this y-axis. With copper 2 sulfate, we can see there is a permanent decrease in maximum rate, and without copper 2 sulfate, the rate is higher. Draw on figure 1.4 to show how you could derive that the Km value for the catalase without copper 2 sulfate. So Km value without copper 2 sulfate will be half of the original value. So we can see 2, 2.5, 2.6. 2.6. So half of 2.6 is going to be 1.3. So 1.3, and then we're going to measure. So we're going to label these as km. And this particular line, this was our Vmax. And for this is going to be our half Vmax. Now the student concluded that copper 2 sulfate was a non-competitive inhibitor of catalase. State the evidence in figure 1.4 that supports this conclusion. If we use this as our Vmax with the inhibitor present, then half of this is 1.4, half of this is 1.2, 1.4, half of this is 1.2, so 1.4, sorry, my bad, half of this is 0.6. So at 0.6 we can see that at 0.6 the km is the same value. All right. Okay guys, so to understand the effect of a non-competitive inhibitor, we have to understand what basically happens with a non-competitive inhibitor. Generally, with the presence of a non-competitive inhibitor, the Vmax itself is going to be lowered. It's going to plateau at a much lower level. However, when we are going to find out the half Vmax of that one, and then we're going to find out the Km value, we will see that the Km value will stay relatively constant. So that indicates that this is a non-competitive inhibitor of catalase. So copper 2 sulfate is a non-competitive inhibitor of catalase. Guys, question number 2a. Inheritance of a petal color and pollen shape in sweet pea plants are controlled by two genes. The gene capital P controls petal color. Allele for capital P is purple and dominant to allele for small p, which is red petals. Gene for capital G controls the shape of the pollen grains. Allele capital G for long pollen grains is dominant to allele small g for round pollen grains. Figure 2.1 shows the structures inside a flower from a sweet pea plant. So petals. We can see the stigma of a female, anther of a female, ovary containing female gametes. Sweet pea plants are normally self-fertilizing. Okay, so they fertilize with their own pollen. Pollen grains containing the male gametes are produced in the anthers and released into the flower. The pollen grains fall on the stigma in the flower, leading to fertilization of the female gametes. A plant breeder cross-fertilized two sweet pea plants A and B. A homozygous dominant plant with purple petals and long pollen grains was crossed with homozygous recessive plant with red petals and round pollen grains. The breeder transferred the pollen grains from the flower on plant A to the flowers on plant B. Suggest one way in which the breeder could prevent self-fertilization of plant B. Okay, so obviously this, generally a female part and a male part, they develop, on a different time. So either the female part can be removed or the male part can be removed from one of the plants. So we can remove the anthers from plant B. Suggest one way in which the breeder could transfer pollen grains from an anther to a stigma. The plant breeder then collected the seeds from plant B, grew the seeds in a glass house. The environmental conditions were standardized. State three variables that should have been standardized in the glass house. Alright, so basically, since we are trying to grow plants, we must definitely have same light intensity, amount of water, carbon dioxide concentration, and other things that we can mention. These are growing medium and also plant density. All of this can affect the growth of a plant, so I'm just gonna add them. Guys, these extra points are there. If you forget these other 3 points, then you can go for this particular 2 points and still get your full marks. Part C. All the F1 plants grown from the seed collected from plant B had purple petals and long pollen grains. The plant breeder then carried out a test cross by crossing the F1 with the homozygous recessive plant with red petals and pollen grains. Alright, so we can see the offspring genotypes, we can see the frequency. Now the plant breeder expected the test cross to result in a 1:1:1:1:1 phenotypic ratio. Guys, whenever we are expecting a particular ratio already, we do not have to work on this particular genetics and find out what actually the original genetics is supposed to dictate. We'll just continue on what they are expecting. Alright, expected and observed. This is our observed. Remember that this is our observed. We are observing this, and expected is what they are expecting. So since we already know the observed, we already know the expected, so observed minus expected should be 102 minus 62. We're gonna then square it and then divided by expected, which is 62, and whatever the answer comes. The null hypothesis of this chi-square test is there is no difference between the observed and expected result. The critical value of P at 0.05 is 7.82 and at 3 degrees of freedom. Use your calculated value to chi-square to explain whether the null hypothesis should be accepted or rejected. Definitely the null hypothesis is rejected. Alright, and why? Because it is greater than the critical value. Our critical value is at 7.82, so definitely rejected. Now the next question says that suggest a conclusion that the plant breeder could make about the inheritance of the genes controlling the petal color and pollen grain shape in sweet pea plant. Alright, so in terms of this particular answer, there is a significant difference between the expected and observed values. We can say this first. The genes are on the same chromosome, so they are autosomally linked. That can be the second point. Part D. Some individuals of the plant species Mimulus guttatus show copper tolerance as they are able to grow on land heavily polluted with copper. Copper tolerance is controlled by an allele of a single gene. Plants that do not have this allele are sensitive to high concentrations of copper 2 ions in the soil and show poor growth. Scientists wanted to investigate if pollen grains are sensitive to copper 2 ions. Pollen's viability is the ability of the pollen to produce male gametes that can fertilize a female gamete. The scientists investigated the viability of a pollen grain by placing them in a culture medium. Normally, after several days of pollen grains start to grow, pollen tubes grow down into the ovary to carry the male gametes to the female gametes. pollen grains are collected from the copper sensitive and copper tolerant plants of M. guttatus, and pollen grains were placed in culture media as shown in table 2.3. Type of plant: copper tolerant. Culture medium containing no copper ions. Number of pollen grains: 200. Culture medium containing copper 2. Number of pollen grains: 200. Okay. All other variables are standardized. Okay, we can see that. The results are shown in figure 2.2. No copper 2 ions. Copper sensitive plant. No copper 2 ions, and we can see percentage of pollen grains that grew pollen tubes is very high. Okay. And then with copper, we can see that's like really low. Those that are copper tolerant, with or without copper ions, we can see the percentage growth of pollen tube is very, very high. The scientists concluded that the presence of copper 2 ions has a significant effect on reducing the viability of pollen grains. Explain whether or not the scientists are justified in this conclusion. Well, we can see that in the copper tolerant plant, there is no significant difference because we can see the the CI overlaps. All right. The error bars overlap. The standard error bar, we can see there is an overlap. However, with copper sensitive plant, yes, there is a significant difference. So guys, that's all for today's video. If you want videos like this, guys, please subscribe to our channel and let the channel grow, and please comment what kind of question paper that you want us to solve. Obviously it has to be related to chemistry and biology. All right, so see you in the next video. Bye bye.